going to get started here. So we are entering our new unit here on genetics. Oh, there are for sure Punnett squares in this, Nicole. You sadly don't have to do quite as many Punnett squares on the actual AP exam, but you do need to know how to do them and interpret them. And there is some math you need to remember how to do from previous units. So I do also have that in these slides at the end, which we'll go over. Our new unit, genetics or heredity, we're going to first start off with traditional Mendelian genetics founded by Gregor Mendel. And then next week, we'll move into non-Mendelian genetics, which talks about different patterns of inheritance that are not quite as cut and dry or simple as Mendel initially observed. And then we will talk about pedigrees as ways to kind of predict and map out how traits are inherited in a family and ways to predict allele frequencies in our gene. Okay. So these notes are on Canvas. We are not using Nearpod today because there are a lot of slides. And some of the slides, I'm going to work out examples. So I wanted to make sure that I could do that here. So let's start by going over Gregor Mendel and his P's that Eli is so excited about. Gregor Mendel was a monk. He worked for a church, lived in a church, and he studied the genetics of pea plants. So he was actually a scientist who also, though, was religious, and that's unique for his time. And he wanted to research and study different types of plants and organisms, but a lot of that was kind of prohibited by the church, right? Because there was a fear that science clashed with religion. So what Gregor Mendel actually initially asked for was to grow a garden of produce at the church for all of the monks and the priests. And cleverly, while he was growing all different types of fruits and vegetables, he used that experience of growing different types of fruits and vegetables to observe genetic traits. So he noticed when he planted his different fruits and vegetables, and peas were his primary plant, and we'll talk about why. He noticed that there were some traits in the plants that seemed to just always show up. They showed up in the majority of the plants, and they were just always showing up. Some traits he noticed seemed like they were almost like hidden or secret, and they just randomly, seemingly out of nowhere showed up, and he called those recessive. And the ones that always showed up were called dominant because they kind of dominated the feature for that. He realized that these traits had to come from the parent plants. They didn't just show up out of nowhere. And then probably one of the more important ones, he noticed that you cannot just tell by looking at an organism which variations of genes it has. And that's important because a lot of times, you know, even students will ask me, how come I can have two parents that look like this and a kid that looks like this? Well, because just because parents look a certain way doesn't mean that they don't have a hidden recessive variation. And I will wait a second for any of us to catch up here. We have quite a few lingering children in here, huh? This class. Oh, goodness. Un momento. Essence! You're good to go. Lo siento. All right. Does anyone need more time on this slide? It's okay if you do. Okay. Okay. So the reason Mendel actually favored pea plants for his studies was because pea plants have such a wide variety of characteristics that he could study. Everything from the shape and texture of the seeds, the color of the inside versus the outside of the seeds, the colors of the flowers, where the different flowers and leaves were located along the stem of the plant, the shape of the pods, the color of the pods, how tall or short the plants were all genetically unique characteristics. And so it allowed him to really study a wide variety of details just by growing peas. So we now fully understand this, right? That offspring are produced from parents by the fertilization of sperm and egg. The sperm, we know from meiosis, comes with half a set of genetic information along with the egg that comes with its own half set, so that when the two combine and fertilize, we get a zygote that has a full set of genetic information. We know that the law of segregation that happens in independent assortment help diversify and get our unique gametes that we need. So we talked about the law of independent assortment, which tells us, remember from meiosis, that each sex cell gets a complete random combination of variations of genes. Okay, you don't have all the dominant going one way when it gets divided and recessive another. You don't have all of mom's variations going one way, dad's variations going another way. Everything gets randomly split up, so that helps guarantee that each sperm and egg cell are unique. Now, a new law we're introducing is the law of segregation, and this also applies to anaphase 2 of meiosis. And the law of segregation just ensures and makes sure that each sperm or egg cell produced only have one variation for each trait. So only a dominant or a recessive variation. And that's how we get the half genetic information. Can the sperm and egg cells be damaged and lose genetic information? What do you mean by lose? No, so likely if the sex cell is damaged, it likely is just not capable of being used during fertilization. Different various things can affect the damage of sex cells. It could be genetically inherent in your body. It could be different types of chemicals you put in your body. So we do know that like smoking, including marijuana and cigarettes and vaping, excessive alcohol consumption, drug use, and that could just be excessive pill use as well, not necessarily, we don't have to think like heroin immediately, can all increase the chance of defects in sex cells, along with damage to their actual integrity. So those things can happen, right? We also know like, especially in males, like restriction to the area where sperm are produced, Excessive heat in that area over long periods of time can damage the production. And that's unique to males as well, right? Because males are throughout every day, producing new fresh sperm, whereas females, we just kind of have like one grocery store trip worth rotting inside of us. That sounds so terrible. It's kind of true. Right, does anyone need more time on this slide? I'll take that as a no. So when Mendel studied his pea plants, and these squiggly plants here are actually pea plants, what he would do is he would start off with two parents that he thought were purebred. So purebred means they only have one type of variation in them. They aren't a mix of variations of traits. So he would start off with parents that one was purebred for dominant, which we'll talk about that, and one was purebred for being recessive, and he wanted to see what would happen. Now, initially, before his experiments, he thought if I mix two purebred parents, the offspring will come out like an in-between. So if I'm mixing a purebred tall parent with a purebred short parent, all the offspring will be like medium height is what he thought. And that's what most people thought. And they thought that's also why, kids look like a blend of two parents, okay? And he was surprised to see that in his first generation, first set of offspring, they all were tall. They all, in fact, looked identical to the tall parent. Well, that was weird, he thought. So does that mean that they're purebred, like you only get your genetic info from one parent? Or what was happening here? So he decided then to breed two of these first generation offspring plants, and these were the results he got. 3/4 of the plants appeared to be tall. 1/4 appeared to be short. Now, this was a huge moment for Mendel because this one, told us that you can't just look at parents or organisms and know what their offspring are going to look like, right? Here, these two parents were tall and they still were able to produce short offspring. So that must mean these parents have whatever variation needs to be inherited to be short. They must have it. They look tall, but they must have that short variation in them. So we know that traits come in variations. This also tells us that you can't tell by looking at them what variations they have. And as we're going to move on to learn that organisms inherit two variations for each trait. That can make them heterozygous or homozygous for the trait. Okay. So again, the first generation he started with were the parent generation, the first plants that were bred. The first group of offspring were the F1 offspring or generation. It's just first filial or first offspring group. F2 was the second generation of offspring that came from it. And that's when he really started to notice some of these unique properties of genetics. So generally speaking, we'll get to this next week, but generally speaking, darker pigmentation and bigger is dominant, generally speaking, okay? It doesn't mean every single trait. And there is no gene for like body height, okay? So it's not like if you're short, you inherited the short variation for body height or something like that. But generally speaking, bigger and darker is dominant. right, does anyone need more time on the slide? Guys, I just want you to know that I am so freaking hungry. Well, I have kids in my room, and so I have to have my mask on. But I want to eat so bad. And just a snack. Just a little something. I do have the wall-up bottle. Tortilla chips. Do you ever dip the tortilla chips in anything, or you just eat the tortilla chips? Yeah, I do have the wall up, and I am vaccinated. But there's a part of me that feels like, just out of respect for the children, you know, they all have to wear masks in my room, so I feel like I should wear a mask. That is true, Nicole. When class is done, I really do sit here and chill and eat for like half an hour. We're done. Spicy salsa, delicious. So how do we inherit these traits, and what do we inherit? Because it's not just pea plants that this applies to, it's also us as humans and all other creatures on this planet. You inherit 2 alleles for every gene. A gene is a region of DNA that codes for a specific trait. And these variations are called alleles. So my example I have in my picture here is that if the gene is for flower color, one variation or allele might be for purple flower color, and another variation or allele might be for white flower color. When you inherit these traits, then, you either are inheriting 2 dominant variations, 2 recessive variations, or a dominant and recessive variation. If you inherit 1 dominant and 1 recessive variation, we consider you to be heterozygous. If you inherit 2 of the same traits, you're homozygous. So 2 dominant traits would be homozygous dominant, and 2 recessive traits would be homozygous recessive. So you're going to notice here in genetics, there's just a lot of vocab. I wouldn't say that the vocab is necessarily very difficult, but it is just a lot of terminology. We're going to talk about that on the next slide. Now I'm thinking about tortilla chips. Oh my God. All right, does anyone need more time here? Oh, JK. Well, congratulations, Valentina. Glad you got a job. That's exciting. Yeah, yeah. Downside. Yeah, I love Panera, but there's actually not as much as I can eat there as I used to be able to because I can't eat meat. So like sandwiches mostly are out of the way. Vinegar, I can't have that, so I can't have salads. Okay, Brian. Yep. So I was actually telling an old student, if you or someone, well, someone at home, you guys are fetuses. If you have a membership to Costco, Costco sometimes sells in the like refrigerated area, kind of by the meat and produce. These big containers of broccoli cheddar soup, and it's the Costco Kirkland brand, but it tastes just like the Panera brand. Delicious. I also love sourdough, Fado. I love sourdough bread. Oh my god. Love it. Their baked potato soup I also really like. I know, we're just making ourselves hungrier. All right, so I'm going to assume we are good. So now let's talk about what an actual dominant and recessive trait is. So dominant, again, is called dominant because if you inherit a dominant trait, it's going to show up, right? That's what it means to be dominant or to dominate a situation, right? You kind of take over. Well, a dominant trait does the same thing. If you inherit a dominant variation, it's going to show up, period. We represent dominant traits with capital letters in genetics. A recessive trait only shows up if the organism only inherits recessive versions. So like Brian was getting at, these can kind of tend to appear to be hidden or show up less often in a population. We represent a recessive trait with lowercase letters. So if I look at the picture on the right, they have set up here something called a Punnett square, which we're going to talk about. We have one parent's traits at the top and another parent's traits on the side. I see each parent has one dominant variation. I know it's dominant because it's a capital letter, and one recessive variation. So if they have one dominant and one recessive, what word would I use to describe the parent? If they have one dominant and one recessive, what word would I use to describe the parent? Heterozygous, good. So both parents are heterozygous. So in this case, the dominant trait is purple. So both parents would appear to be purple. And if they were to have offspring, there is a 75% chance that their offspring would look purple, but there is a 25% chance that their offspring would look white. And so that's kind of the discovery that Mendel didn't see coming, right? You see purple flowers, you think they make purple flowers. But in fact, flowers could look purple and carry a gene for a totally different flower color. We see here in the bottom right corner, this white flower looks like the recessive white trait because they inherited only the recessive variation. Again, if a dominant variation is inherited, it's going to show up. So in the case in the diagonal here, where I have two heterozygous offspring, they might have the lowercase p for the white trait, but they aren't going to look like that. They just have it in their DNA. They're always going to look like the dominant trait if they inherit the dominant trait. Really good. Okay, so if we look here, again, another way to break down. Here we represented our dominant trait again with capital letters, and here it's capital B's. The recessive variation is a lowercase b. So again, if we look at the homozygous dominant and heterozygous versions of these flowers, both look purple, but the variations they inherited are slightly different. One has only dominant purple variations, but the other one has a dominant and recessive variation. Remember, no matter if it inherits one recessive variation, if it inherits a dominant variation, it's going to look like the dominant variation. So heterozygous organisms always look like the dominant variation. Again, homozygous recessive, you only look recessive when an organism is homozygous recessive. They only have the lowercase recessive variations that they inherited. So there are two different ways then we can describe the traits that you inherit. We can describe the traits by how they look, how you look, or by the actual alleles and genetic information. If I describe an organism or a trait by how it looks, I am describing its phenotype. So phenotype describes a physical appearance of a trait. So phenotype and physical. So to say something that is white, brown, purple, white and brown spotted, tall, short, those are phenotypes because with my eyeballs, that's what I see. If I get into detail and describe the actual variations of a gene that was inherited, I'm describing the genotype. So genotype is the genetic description for that trait. So to say that an organism is homozygous dominant, heterozygous, or homozygous recessive is describing the genotype of that organism. All right, does anyone need more time? Alrighty. So let's make sure we clarify these letters again, because it's going to be very important as we move forward into our Punnett squares. The letters represent the alleles an organism can inherit. If an organism is homozygous dominant or inherits 2 dominant variations, I would use 2 capital letters. If an organism is homozygous recessive or inherits 2 recessive variations, I would use 2 lowercase letters. And if an organism inherits 1 dominant and 1 recessive variation or is heterozygous, I would write 1 capital and 1 lowercase letter. Also, a heads up, you are going to need a coin for class Thursday. No, it just needs to have a distinguished head and tail side. Sounds wonderful, Valentina. All right, are we good? Okay. So here's what we're going to get into next with Punnett squares. Again, you can see we have our letters. I put 1 parent at the top, 1 on the side, and the variations of the gene that they inherited. Our boxes then on the inside are going to represent the possible offspring they could have, okay? And it's specifically the possible types of offspring based off whatever trait I am making predictions about. So how do we predict inheritance? Well, we use a tool called a Punnett square. And Punnett squares, again, tell us the probability of offspring having certain traits. Punnett squares are not psychic. They don't predict the future. They don't tell you that these two parents will have 4 offspring and that's what each offspring will look like. It's probability. So as we break it down, we're going to look at each box represents a 1 in 4 chance or 25% chance of having offspring that inherit those variations of traits. Only think about food. Yes, Valentina. So Valentina asked, are things like this used when a doctor tells someone your child has a blank % probability of getting genetic disorder? Yes. So, well, let me clarify. Mostly yes. If we're just basing it off something like the mom, her age, any other health issues she may or may not have, the % probability then is statistics based off like nationwide and international data points that are collected. However, if it's between 2 parents and we are following a trait through the family, then yes. Okay, does anyone need more time on this slide? Okay. So how do we set up a Punnett square? So when we set up a Punnett square, one parent's genotype goes on the top of the Punnett square, and another parent's genotype goes on the side. It doesn't matter which parent goes where, just one and their variations goes on the top and another and the variations on the side. So here I use colors to help distinguish that. If one parent is homozygous dominant, so 2 capital A's, you see I have that one parent here on the top and I put 1 variation per box. If the second parent is homozygous recessive, notice in the lime green here, I have them on the side, 2 lowercase a's, and again, 1 letter per box. You're going to see as I work through a Punnett square problem with you as well that my Punnett squares are more like open Punnett rectangles. I just like more space to write. Whoa. No, Angel. So the dominant variation is one physical expression of the gene, and recessive would be a different. I kind of think you're maybe getting at something we're going to talk about next week, but I'm not going to bring it up this week. All right. So how do we fill in a Punnett square? Well, they fill them in like a matrix box or like a grid on a map. You're going to take the letters at the top and copy them straight down into all the boxes below. You're going to take the letters on the side and copy them straight across into all their boxes. You always want to make sure that you write the capital letter or the dominant trait in the front. And otherwise, that's it. That's how you fill them in. And we are going to do one example live together. Yes. So Valentina said, so in this case, there's a 100% chance that the offspring would be capital A, lowercase a, or heterozygous. Yes. And this is actually what Mendel found in his first parent cross and first offspring generation, right? The parents were purebred, meaning homozygous. One was homozygous dominant, the tall plant. One was homozygous recessive, the short plant. And he thought again that he was going to produce offspring that were a blend of the two, because at that time, that's what they thought happened. You were a blend of traits from one parent and another. And so he thought they were going to be this unique, like medium type height. And instead, he saw all tall plants, just like we see here. He didn't know at the time, though, until that second cross, right, that even though all these offspring could look tall, they could kind of like secretly carry this recessive variation that could be passed on to future offspring. And so that was a big turning point again for letting him know that even though an organism looks one way and it may be dominant, that doesn't mean that's the only variation they carry. Okay, does anyone need more time? Okay. So how do we interpret a Punnett square? Well, each box is a 1 out of 4 or 25% chance. So here we would say we have a 4 out of 4 or 100% chance of offspring being heterozygous, or a 4 out of 4 or 100% chance that the offspring would look dominant. Yes. But I do want to just say, yes, genetics works the same for all organisms. However, different types of organisms do have different amounts of genes or traits and different numbers of chromosomes. And next week, we are going to talk about that while a lot of our traits do follow this basic Mendelian set of principles, as we develop more technology and learned more, we also learned that there's a variety of traits that don't just follow these basics. Well, remember that Gregor Mendel actually initially studied pea plants. All right, does anyone need more time on this slide? Okay. So we are going to do a little Punnett square problem here. It's not a difficult one. I feel like there's a fuzzy inside my mask. It's driving me bananas. Okay. Let's see, is this gonna work okay? Not really. All right, it says we cross a homozygous recessive purple organism. So one parent is homozygous recessive purple. Okay, with a heterozygous white organism. One other parent. Oh my, why? Why do you hate me? Is heterozygous. You know, I always feel like I need to defend myself. I swear, if I'm writing on a whiteboard or on paper, my handwriting is significantly neater. And so when it says we cross them, that means we mate them or have them reproduce. Okay, so in order for me to do a Punnett square here, I need to figure out their genotypes, right? Which trait is dominant, purple or white? In this scenario, which trait is dominant, purple or white? Good, it is white. How did you know white was dominant? You are correct, it is white. How did you know it was that? There we go. So if it is heterozygous, right, oh my gosh. If it is heterozygous, that means that there is a capital letter and a lowercase letter, right? And if it looks white, that means it looks like the dominant trait. So if white is dominant, then purple must be what? Recessive. Okay, they also tell you that here. And so to be homozygous, recessive, and purple, I would be 2 lowercase letters. Now there might be some of you sitting here thinking, well, why did you use A's? Well, the letters you use don't actually matter. I tend to always use A's. It is easy to distinguish between the capital version and the lowercase version. It doesn't confuse anyone. You know, I don't ever want you to think like, oh, I'm gonna write big W, little w for white and 2 little p's for purple. I need to pick one letter and stick with it. Okay, so that's why I just always pick A's. The letter itself is not what matters. It's if it's capital or lowercase and how I interpret it. So now I'm going to use those to make my Punnett square, and I can make some predictions. So again, my Punnett squares, because mostly I'm lazy, are open Punnett rectangles. One parent is going to go at the top, 1 letter per box. And the other parent is going to go on the side, 1 letter per box. Are we okay so far? All right, so then I'm going to take the letters and I'm going to copy them down into the box. I always want to put the dominant traits in the front, so it's the left side. So big A, lowercase a, lowercase a. And then I'm going to go from the side, copy them over. And that is my completed Punnett square. Okay, does anyone have a question so far about that? Okay. So if I look at this Punnett square, what are the chances of these 2 parents having offspring that look dominant? All right, so 2 out of 4, 50% chance, right? That would be these 2 boxes right here. Okay. What are the chances of these 2 parents having offspring that look white? Ah, 50% chance again, because remember, white is dominant. Okay, what are the chances of these parents having offspring that are homozygous dominant? Good, 0 out of 4, 0% chance, because that would be 2 capital letters, right? But what are the chances of these parents having offspring that look purple? Okay, 1/2 again, right? So here and here are purple ones. They're homozygous recessive. So 2 out of 4, 50% chance. Okay. So again, it's not saying that 50% of the offspring will look purple. It is saying every single time these parents conceive, there is a 50% chance the offspring could look purple. Okay. All right, any questions on that? All right, now, those are what we call monohybrid crosses, where we only focus on one variation. What I'm going to show you next is slightly terrifying. I'm not going to sugarcoat it. There are dihybrid crosses. Dihybrid means we are looking at two variations being inherited simultaneously. Okay, so two different traits and their variations inherited at the same time. It looks like this. These are not common. College Board says you don't have to do these on the exam, but yes, Bryce, it's been 6 hours, yeah. Yeah. There is a problem where you have to do a dihybrid Punnett square to figure out a percentage of something. So you have to, but you don't have to. So I have some examples in here. It can be one of those things that we go over in class an upcoming week or something, or when we have extra time or on a Friday. Again, it's not something common. You don't have lots of problems in your homework and you won't have lots of problems on the exam. I think there's only one question that I could find in College Board's practice questions that even involved a dihybrid cross. And it's not that they're asking you to you're not getting graded on the setup and fill in of it. You're getting graded on the interpretation of it. So you have to do it, okay? You also are going to have to remember chi-squared, degrees of freedom, and finding critical value, okay, or you're using your p-value here. So I did put these slides here at the bottom on how to calculate these things. So here's the equation for chi-squared. The table that you get for that actually gives you your observed and expected frequencies. So you just have to plug and chug. Remember, this weird symbol represents the sum of, and a sum is just adding stuff up. Degrees of freedom, this is how you calculate it. And this is how you find critical value, okay? %, Brian, which is also why we are not allowed to genetically modify humans. So when they talk about genetically modified food in general, or you see like non-GMO means that the animal, right, the plant or the animal that was used for that food is not genetically modified. And what that means is we genetically modify almost everything we eat. So if you eat any non-organic anything, it's guaranteed to be genetically modified. We genetically modify fruits and vegetables and animals to be larger, to have more fruit on them, to alter the flavor of them, to alter which types of climates they're able to survive in, which is why we're able to grow so many of the different fruits we do in the United States year-round. We can genetically modify them to be poisonous to certain insects, but not humans, so a natural form of insecticide. And the reason I would say a good number of people can be anti-genetically modified food is because there's a worry that we don't know what the long run of that is in our bodies. I don't think that's anything to be worried about. When I eat a plant, their DNA doesn't fuse with my DNA, so it doesn't really matter. I essentially poop it back out. But some people don't like that idea that we are consuming food that is not how it would be in nature. But yes, 100%, we do use that all the time. Okay, so your homework assignment is the practice that goes with this. So down here at the bottom, you have your 6.1 practice. That's going to be due the end of this week. We're going to do a little activity in class Thursday. I think it's fun. And I do have your supplementary videos here. We only have about 10 minutes left of class, so it's up to you. If you want me to show some of these videos in the last 10 minutes, I can. If you would like to just leave early and take care of your business, you are welcome to do that as well. What would you guys like me to do? Don't feel offended either way. So then I'll just say this. Oops, okay. Let's go to, well, you guys, I can't see you guys. Okay, let's do monohybrid process here. William, you are good.
So I have a confession to make. For my classroom, I always wanted a hairless guinea pig. They sell them at some pet stores, but they can be really hard to find. And while I doubt they do so well in the wild, their genetics are fascinating. And so are guinea pigs. We spend quite some time on LOL cats, but we'd spend even more time if someone would make a funny guinea pig site. Most guinea pigs have hair. This is because of their DNA. DNA makes up genes, and the guinea pigs received genes from both their mother and their father. An allele is a form of a gene and often represented by a letter. In this case, we'll use the letter H for hair. A hairless guinea pig has two recessive alleles for the trait of having or not having hair. A recessive allele is usually represented by a lowercase letter or allele. By recessive, it means that the allele will not usually show up. The only way it will show up is if there is no dominant allele present. A dominant allele is represented by a capital letter, and it's an allele that will show up. Think of it as dominating. So a hairless guinea pig does not have a dominant allele for hair, and that's why the recessive trait, not having hair, shows up. A hairless guinea pig's genotype, that's the genetic makeup of an organism, can be represented as a lowercase little h, little h. A guinea pig that does have hair can be represented as a big H, or a big H, little h. See, it only takes one dominant allele, a capital H, for a trait to show up. That dominating allele means that the recessive allele is hidden. Now, we're not quite done with the vocab here. A genotype of big H, or little h, is considered homozygous. The root in this word means same, and they are the same case. Big H, those are both capital letters, and little h, those are both lowercase letters. So big H is called homozygous dominant because of the capitals, and little h is homozygous recessive because of the lowercase. A genotype of big H, little h is considered heterozygous. The root in this word means different because their letters are different cases. One is a capital and one is a lowercase. If I were to show you a guinea pig with hair, would you know its genotype? Well, you would know it's not little h, because if it was little h, then it would be hairless. But remember, a guinea pig with hair, it could be big H, or big H, little h. We don't really know, When we could do a test cross to eventually determine this, but you can't tell just by looking at the guinea pig. Now, if we had a hairless guinea pig, we do know for sure that it's little h, because if it did have a capital letter, even one, that dominating allele would take over and it would have hair. So let's try a monohybrid cross. The root mono means one because it focuses on one trait, in this case, hair. And that means a Punnett square would need to be created like this with 4 squares. In our cross, we are going to cross 2 heterozygous guinea pigs. So let's look at this in steps. Step 1, first, figure out the genotypes of the parents. Heterozygous means big H, little h. Step 2, place one parent along the top of the Punnett square, outside of the boxes like this, and place the other parent along the left side of the square, outside of the boxes like this. Step 3, cross them. For formatting purposes, we always put the capital letter first. The results that you get in the squares would be the offspring, the babies. Now, if I were to ask you the genotypes of the babies, you could list them out. 1 big H, 2 big H, and 1 little h. I could even turn that into a genotype ratio of 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive, or a percentage. 25% are homozygous dominant, 50% are heterozygous, and 25% are homozygous recessive. Well, what if I asked about the phenotypes? What does that mean? I like to think that pheno sounds like physical. So the phenotype would be the physical traits of that organism. And in this example, it's whether they have hair or not. Now remember, any babies that have a capital H have a dominating allele, and they will have hair. So the babies that are big H, or big H, little h, they have hair. So 3 of them here, the 1 big H baby and the 2 big H, little h babies, they all have hair. The homozygous recessive baby, which was little h, that baby has no dominant allele present. So this little guy would be hairless. So you could say the phenotype ratio is 3 have hair to 1 that's hairless. Or a phenotype percentage could be 75% hair, 25% hairless. One thing to emphasize about Punnett squares is that they are predictions. These are probabilities. This means it's not necessarily exactly what you're going to get. For example, it's a probability that a child has a 50-50 chance of being born a boy or a girl. But we all know families that only have children that are girls or families that only have children that are boys. Probabilities are predictions. Another reason to love biology, it's exciting. Well, that's it for the Amoeba Sisters, and we remind you to stay.
Okie dokie. So don't forget to check out with your advisory teachers, and the recording for this eventually will be linked to your biology slides, okay?